Hello everyone, my name is Henry Valère and I would like to start by thanking the organizers for giving me the opportunity to present my work, ChromFormer, which is a transformer-based model for 3D genome structure prediction. Now, we aim to predict the 3D genome structure, and this is important because the genome structure affects if, when, and how genetic information is expressed. And a disruption in this conformation has been linked to various cancer types, neurological problems, and diseases. However, compared to proteins, well, there are no experimental methods to find the 3D genome structure. The only insight is given by high-throughput chromosome conformation capture techniques, also called HIC. This technique creates a matrix that contains the interaction frequencies between parts of the genome. Various methods use HIC to predict 3D chromatin. Among the most popular are distance-based methods and contact-based methods, which our method is part of. Distance-based assumes the existence of a transfer function that underlies the relationship between interaction frequencies and 3D Euclidean distances. However, the transfer function needs to differ across different organisms, chromosomes, and resolutions. Contact-based methods directly map the interaction frequencies to an output structure by assuming that the lower dimensional structure is embedded in the higher dimensional HIC. However, contact-based methods architecture is not designed to generate encoding resembling 3D structures. As existing methods are unsupervised and limited by underlying assumptions, I here present ChromFormer. The first problem is the lack of ground truth and data. So we first use ClusterWalk, which is a novel synthetic data generation module that simulates biologically feasible structures, as seen here, and corresponding HIC matrices, as seen here. For HIC to resemble real HICs, we use processing steps, including normalization and optimal transport. The created HIC is then fed to ChromFormer, which predicts the 3D genome structure and the local confidence. We optimize the model using three objectives. The first two are distance-based, losses between predicted and true structures. The third one is a confidence loss, which is simply a variation of AlphaFold's local distance difference test. Now, our framework was tested on the TrueSalt dataset, which is an independent synthetic public dataset containing 100 simulated structures paired with a single HIC matrix. Using a ClusterWalk algorithm, we generated 1,000 structures that match the TrueSalt dataset as seen here. We as well generated HIC matrices that resemble the TrueSalt HIC, as we can see here. We then evaluated our method using the capture distance, where we can see that ChromFormer outperforms state-of-the-art methods, not only in numbers, but as well visually, where we can see that ChromFormer's outputted structure resembles the most the TrueSalt structure and is the most biologically feasible. Now, our method predicts as well confidences, and we observed that the calibrated confidences with isotonic regression were the ones that were the most accurate. We can see the calibrated confidences in this example. To summarize, we created ClusterWalk, which can easily be used to generate data, and this data is used to train ChromFormer in order to predict 3D structures as well as confidences. Now, the main problem though with our method is that for each new tested dataset, new data needs to be generated, which can be computationally intensive. I would like to thank the rest of the team at ETH and IBM Research Europe, Zurich. Thank you very much for listening, and bye-bye.